Hi guys, welcome to this short video on immunology questions. I've tried to pick some, you can say high yield questions, especially those questions that I have not covered in my other videos on immunology. So this will just help you kind of complete the whole picture. Okay, so let's get started. Question 1. Which portion of an antibody provides antigen binding sites? And pause this video and try to answer this yourself, and then unpause when you're ready to hear the answer. Okay? So it's the variable portion. The variable portions are up here, and these are the ones that bind to the antigen. Okay? And this is my constant portion, which binds to my immune cells. So my neutrophils or my eosinophils would bind here. Okay? And this is a slide that shows you how we really put together this antibody. Okay, how do we really make this antibody? Remember, our antibody, the light chains are called the kappa or the lambda chains, the heavy chains are called the H chains. And this picture here shows you how we make them and how we kind of arrange them and how recombination takes place. So what you're looking at here is the mRNA. The mRNA here with the blues, this is the mRNA that will help us make our heavy chain. So in the heavy chain mRNA, you can see we have V, D, and J regions. So the variable diversity and joining regions, we have V, D, and J. And we get V, D, J recombination when we are making our heavy chains. In the mRNA for my light chains, we only get V and J regions. So we only see V and J regions, so we only get VJ recombination when we're trying to make our light chains. And who's kind of controlling this whole thing? It's RAG1, RAG2. RAG1, RAG2 is really what's allowing the recombinations to take place. Okay. Question 2. Which of the following correct option interacts with CD4 positive T cells to elicit an antigen-specific adaptive response? So pause, and once you're ready to hear the answer, you can unpause. And the answer is B, MHC2. Remember, MHC2 is what pairs with our CD4 or antigen presents to our CD4 positive T cell. So MHC2 presents antigen, is on the antigen presenting cell, and it contains 2 alpha, 2 beta chains, and I'll show you a picture in a second. And if you want, you can pause here and just read the differences in both of them. So MHC molecules, we have class 1 and class 2. The way I like to remember this, class 2 has 2 chains for each. So there's alpha 1, alpha 2, there's beta 1 and beta 2, right? And the alpha 2 and the beta 2 chains are the ones that go through that plasma membrane. In class 1, I only have 1 beta chain, and the beta chain, if you see, is not going through the plasma membrane. However, we have 3 alpha chains, alpha 1, alpha 2, and alpha 3, and alpha 3 is what goes through the plasma membrane of the antigen presenting cell. Okay, question 3. A 14-year-old female develops hives after being bitten by a bee during her camping trip. An evaluation of her blood would show an elevation of which antibody? So hopefully this is the easy one, right? And the answer is IgE. And IgE, remember, this is the one where we see, with the parasitic infections or allergies, anaphylaxis, those kind of things. It helps degranulate mast cells and cause inflammation. So when you get a bee bite, you see that redness, it swells up, it's hot, it's itchy, classic signs of inflammation. And that's because, mast cells are degranulating, releasing all that histamine. And let's look at the structure of an IgE. The valency of an antibody is how many antigen binding sites it has. So 1 and 2. IgE can bind 2 antigens. So valency is 2. However, it's a monomeric antibody. Okay. IgM, you can see here, it has 10 antigen binding sites. So the valency is 10. And this is a pentameric. It's a big antibody. Okay, it's a big one. IgG is the main one. And I think something really important to remember about this is that this is the only antibody that can cross through the placenta and go from the mother to the fetus. This is again 2 antigen binding sites, so valency is 2, and it's monomeric. And IgA is really important. It's present in our mucus lining, in our throat, in our like upper respiratory. It just helps. Acts like the first line antibody. Okay, it's like the first line agent in the mucous membranes. And it has 4 antigen binding sites. So valency is 4, and it's dimeric. Okay, and you can read through these. You can pause and then read through this whole table when you get a chance. Okay, and this just shows you that there is a diversity. We can make IgM, IgA, IgG, IgE, IgD, different types of antibodies, and it involves a lot of kind of recombination, and we'll talk about this in one of the follow-up questions. Okay, the developmental step that commits a cell to the B lineage is. So think about this, pause your video and then unpause when you're ready to answer. And in order to be able to answer this question, we have to understand the development of a B cell, okay? How a B cell goes from being a stem cell to being a mature B cell. So how you read this picture here first is, so you have, as you go to the right, you have your B cells getting more mature. And on the left, you can see your heavy chains, light chains, and IgG status at each phase. So let's say we're looking at stem cell. Does a stem cell have heavy chains? No, it's still germline. Does it have light chains? No, it's still germline. Does it have an immunoglobulin status? None. The step really to answer the question is, the step that commits a cell to B cell lineage, it's this first step going from stem cell to an early pro-B cell. This is the step that they asked in the question, okay? So to go from here to here, you actually, do you see heavy chain recombinant? Yes. You start to see D and J heavy chains, D and J rearrangement in the heavy chains. Any light chain genes? None yet, and no Ig status. The early pro-B cell becomes a late pro-B cell, and now we're starting to see VDJ. Okay, so VDJ rearrangement. I like to think the DJ needs to come and like start the party, and then the other people gonna join. Okay, so DJ first, and then VDJ rearrangement in the late pro-B cell. Any light chains? Not yet. Any Ig status? None. The late pro-B becomes the large pre-B cell. We have pre-B, we have VDJ rearranged fully. No light chains. However, we have these pre-B receptors that are being expressed on the cell surface. So if you see the Ig status, mu heavy chains is what we see, which are surrogate light chains. Pre-B cell receptors on the cell surface. So remember the pre-B cell. So heavy chain is good, it's ready, but no light chain yet, but we do see pre-B receptors on the cell. Now this becomes the small pre-B cell. Small pre-B cell has VDJ, obviously is rearranged, carries on. And now we're starting to see some light changing rearrangement. So we start to see the VJ rearrangement. And like we had mentioned in one of the earlier slides, I showed you that light chains only have VJ, heavy chains have VDJ. And Ig status is now we have that mu chain in the endoplasmic reticulum. This progresses into an immature B cell. This is important. This is an important stage because now you have VDJ, heavy chain rearranged fully, light chain rearranged fully. And now you also start to express IgM on the surface. You're expressing IgM on the surface, right? And then finally, it's mature B cell where you express both IgM and IgD on the surface. Okay, so make sure you know these, at least focus on the ones that are circled in red. I think they're more tested. Okay, how is this different from T cell, differentiation? So T lymphocyte progenitor cells are in the bone marrow. They migrate to the thymus, okay? So in the thymus, they're called thymocytes. and they migrate towards the cortex of the thymus, and we start to see thymocyte proliferation. So one of the first things we see is thymic stromal cells, cytokine secretion, IL-7 and Notch1. So IL-7 and Notch1 is one of the first things we're going to see for thymocytes to proliferate. Once we have thymocyte proliferation, the next we have is a double negative thymocyte, a thymocyte with no T cell receptor, to put it like that. Okay, so double negative. This double negative thymocyte goes through TCR gene arrangement or T cell receptor gene arrangement. And again, remember T cell receptor gene arrangement, who are going to be the key players, RAG1, RAG2. Okay, anytime gene rearrangement, who's the key player? RAG1, RAG2. Yeah. Okay, and once you do that, you are able to successfully, you have successful TCR beta chain rearrangement. So the double negative thymocyte goes through pTCR proliferation. And once it goes through the step, you essentially go from having a double negative to a double positive thymocyte. Okay, so you go from having nothing on the no receptors to having both CD4 positive receptors and CD8 positive receptors. Okay, so that's a double positive thymocyte that has a T cell receptor for CD4 positive and CD8 positive. So once you're here, you do a positive selection. And a positive selection is one of the most important steps, I would say. When you have a positive selection, you select for either the CD4 or the CD8. So you go from having both double positive to being a single positive, where you select for either CD4 positive or CD8 positive. Okay, so the T cell selects what type of T cell it wants to be. And once it does that, excuse me, it goes through negative selection. Negative selection is we present antigens to the T cells and we see if there's any cross reaction or if it's if it can damage the host cells in any way. Okay, so if the cells can damage host cells in any way, we get rid of them. Okay, those don't mature. Only the good ones, the cells that are very good at getting rid of the infection only, those mature into mature CD4 and CD8 positive T lymphocytes, which are then sent out of the thymus. And this is like a really nice picture for them. You can see we have our double negative, and then immature double positive, and then mature either mature, here it says either CD4 positive or CD8 positive. Okay, so these are some important things you should remember. IL-7, Notch1, that starts the process, gets the thymocytes to proliferate. For TCR gene rearrangement, you need RAG1, RAG2, but you also need CD3. And then you get a TCR that is positive for both CD4 and CD8, so double positive. And then you want to select for only one, so either CD8 or CD4, which you call positive selection. And then you see a negative selection to only keep those that are effective and won't hurt the host, which we call negative selection. Okay, question 5. Negative selection of B cells occurs when, so we just talked about this, right? So hopefully this would make sense. Negative selection of B cell occurs when immature B cells are able to bind self-antigens, and that's when we kind of get rid of them and then they undergo apoptosis. Okay. And this is talking about B cells, and we just talked about T cells, but it's pretty much the same thing. Okay. We also have negative selection in B cells, where the immature B cells that are able to bind to self-antigens, we get rid of them and send them for apoptosis. Question 6, major histocompatibility complex, or your MHC, refers to a large group of genes that codes for proteins that play an essential role in which of the following? Okay, so once you're ready, you can unpause. And this should be easy. MHC help with antigen presentation to T lymphocytes. Okay, and I believe this is the last one. A 5-year-old girl comes in with symptoms of Neisseria infection. Deficiency of which of the following will lead to recurrent infections with an encapsulated gram-negative bacteria? This is, I believe, a really high yield question in terms of like step prep. So a young girl who's getting repetitive Neisseria meningitis infection, excuse me, and the answer is C5. Excuse me. So you have C5. Okay, and that's all the questions that I have for you. Remember that there's someone getting recurrent Neisseria meningitis infections. It's usually a deficiency of complement 5. Okay, and that is all the questions. I do have this little list of some important T cell and B cell markers, and I hope this helps. Thank you for your time, and yeah, thank you.